Bringing a drug into the market is a very long, complex and expensive process. To determine if a drug is safe and efficient, it first needs to be tested. Current models for testing drugs in the lab are either just too simple to accurately model the human body and drug interactions, or, in the case of animal models, often fail in predicting human response. Most of the new cancer drugs are so specific for humans that testing them in animals is almost impossible. At the end of the preclinical phase, the selected drug candidate is then administered to humans during clinical trials. Therefore, these drugs should be as safe as possible with no potential side effects for the patients. Unfortunately, still many drugs fail in clinical trials as they are not safe enough or they don't show the clinical benefit that is desired.
This is why new and advanced in vitro models like the AlveoliX lung-on-chip are needed. The lung-on-chip is a tool to test and evaluate drugs in the biology laboratories in the preclinical phase of the drug development process. The goal is to better predict how a drug is acting on the human lung early on in the process. Inside the lung-on-chip, we can culture human lung cells derived from patients. So we create like mini lungs inside the laboratories. After some time having them in culture, we can let them breathe. The breathing motion is essential to keep the cells in a state as they would still be in the human body. This allows us now to have an advanced model combining chip technology, biology, as well as breathing motion to test drug candidates in an environment, in a model which is very close to the human body.
In the last 3 years, we could show the preeminence of our technology. In cooperation with leading pharmaceutical companies, we could prove that it is very predictive, and we could also show that it has a very translational power for the clinics. In contrast to conventional drug testing models, our technology brings key advantage for the end users, but most importantly for the patient. It will reduce costs and time that is needed to bring a new drug on the market. It will reduce the attrition rate. And most importantly, it will also reduce animal testing, which are cruel and not needed. The patient will be treated with better and safer drugs. The access to the drug will be much quicker, and we can reduce unwanted side effects. Having started now with the lung-on-chip, our team is already developing new mini-organs like skin, gut, brain. The goal is to move towards personalized medicine. Simply speaking, we want to test a new drug on the chip before it comes to the patient to find out the best treatment option for this individual patient.